Howdy guys, welcome to the first lesson of the 5th unit, which is DNA and genetics. We're going to just go over some basics of nucleic acids. This is mostly stuff that you should have learned your freshman year of biology with a few extra things thrown in. So don't discount this lesson. There's going to be some new stuff in it. We're just tying it in with the old stuff. So let's talk real quick about how we know the basic structure of DNA, because there are some foundational experiments that it is helpful for you to know for the AP test. So there are two people who are widely credited with creating the first model of DNA that is molecularly close to what we know as the structure of DNA. That is Watson and Crick. However, they did have the help of Rosalind Franklin, who was a woman technician who created a lot of the X-ray crystallography images that they used. She worked in the lab of Wilkins, who shared those images with Watson and Crick, who used it to then derive the structure of the three-dimensional structure of DNA. So what they ended up doing to create this model was they used a technique called X-ray crystallography. So what they do is they have a source of X-rays that is then shined onto a crystallized sample of whatever molecule you're looking at. So it doesn't just have to be a DNA. And then they have a plate which records which X-rays, which areas have X-rays reached, because the X-rays were diffracted or they were bent by going through this crystalline structure of a molecule. And based on the way in which the X-rays were diffracted or bent, you get this kind of image of like X-rays that have landed onto this photographic plate, and you can then use that to try and figure out the original structure of the molecule that these X-rays were bent by. It's not the easiest technique in the world. However, before we had a lot of the modeling technologies or understood a lot of how the base pairing or how the molecular interactions happened to form the three-dimensional structure of proteins or other molecules, this is how a lot of the structures of these molecules were determined. So from this and from some chemical studies, they learned that DNA contained these two groups of molecules, purines and pyrimidines. What you see is that the purines have a double ring structure. So adenine and guanine are your purines, and notice they have these two rings. They're carbon-nitrogen rings that are fused together, whereas pyrimidines are much shorter. They only have a single ring that makes them up, and there are three different pyrimidines that are found in nucleic acids. Cytosine and thymine are found in DNA, and in RNA, that thymine is replaced with uracil. Something that another teacher told me that she was taught in school to help her remember which bases are pyrimidines and which ones are purines. They were told Aggies are pure, which have A and G, and pure means purines. I also think of it, the longer name, or pyrimidines, goes with the shorter molecule. So pyrimidines have one ring, purines have two rings. What you'll note is that when you have bases pairing, which we'll talk about in more detail in a minute, A pairs with either T or U. Notice that A has two rings, and T and U both have a single ring. So if you'll look at an area of DNA which has an adenine on one side and a and a thymine on the other side, since it's DNA, you'll notice that it's three rings across. Something similar can be seen with guanine, which also has two rings, and how it pairs with cytosine, which has, oh sorry, I forgot to draw the second ring, and pairs with cytosine, which has only a single ring. So notice that you also have three rings across. This is important because knowing that these bases pair up in this predictable way lets you see that a DNA molecule will always be three rings across. It gives it a uniform shape, so if you were able to untwist a double helix, it would be a uniform width the entire way through. So every complementary base pair is going to have one purine and one pyrimidine in it. The base pairing rules were identified by a scientist named Chargaff. He did experiments where he calculated the percentage of each, the relative proportion or percentage of each base in multiple organisms' DNA. So he would look at humans. Our DNA, out of all the 100% of bases, about 30% was adenine, about 30%, close to, was thymine, about 20% was guanine, and about 20% was cytosine. So he noticed that this kind of pattern held true. However much adenine you had, the relative proportion of adenine in the genome of an organism, you would have just as much thymine. That means for every adenine, you would have a thymine. And the relative proportion of guanine in the organism's genome would always be relatively equal to the proportion of cytosine, meaning that for every cytosine, there would be a guanine. And this is how he determined that A and T will always pair with each other, and C and G will always pair with each other. You'll notice that these numbers are not exact, so 30.9 and 29.4 are not 100% equal to each other. However, when you start doing statistical tests to determine like the margin of error of these things, those two values are relatively equal within the margins of experimental error. Because remember, well, we'll talk about experimental error and things like that in more detail later as we get closer to the actual test. So this is showing you the base pairing. You'll see guanine and cytosine will form a certain amount of hydrogen bonds with each other. They will form 3 hydrogen bonds with each other, whereas adenine and thymine will only form 2 hydrogen bonds with each other. The fact that these different numbers of hydrogen bonds that they form is going to be responsible for their particular base pairing. So guanine will not try and pair with thymine because they will form different numbers of hydrogen bonds with each other. This ensures that kind of specificity of base pairing. So that way you will not have guanines pairing with thymines or adenines pairing with cytosines. So this is just showing you the 3-dimensional structure kind of DNA. You know that it has a double helix, which means it's that kind of twisted staircase ladder, with the steps of the ladder being the nitrogenous bases and the sugar phosphate backbone, in this case being represented by a ribbon. Notice we also have these terms 5' and 3'. 5' is going to have a hanging phosphate group that's going to be free, and 3' is going to have a hydroxyl hanging free. And so that just kind of shows you that is involved with the directionality of these molecules, so you can only add at a certain end. DNA and RNA and all nucleic acids are built in a certain direction, and they are read in a certain direction. So they are built 5' to 3', they are read 3' to 5'. So you have these enzymes that will bind on, and they are the ones that will add new nucleotides or will read the nucleotides, and they will only move in certain directions and attach at certain locations. So that's what I said here. All of that information is there. So 5' has a hanging phosphate, 3' has a hanging hydroxyl, etc. DNA strands are complementary to each other, and that means that we can use the sequence of nitrogen bases on one strand to predict the sequence of the other. They will match or fit with each other. They are also anti-parallel, meaning that one of them will run in the 5' to 3' direction, and the other one would be, if you go in the same direction. So here, if we're looking at this strand of DNA, it goes from 5' to 3'. If we start at the, quote, top of that DNA molecule on the other strand, it's going 3' to 5'. So they run in opposite directions to each other. So let's talk about how DNA is packaged within a cell. There is a lot of DNA that gets stuck into a single nucleus. If you took all the DNA in one of your cells out and you laid it end to end, you would find that you have about 6 feet of DNA 6 feet long strands of DNA stuck into the nucleus of a single cell. So this has to be packaged and organized in such a way that it doesn't get tangled because it is very long and thin. If you've ever messed with lots of thread or lots of string, you'll know it's very easy to get tangled up. So what ends up happening is your double helix gets wrapped around these proteins called histones. Then those histones with their wrapped DNA around them also will coil up, and then those coils will form coils, and those coils will form coils until eventually you get to this large structure that we recognize as a chromosome. It is that X-shaped structure, though I will point out that this is actually 2 identical chromosomes joined together. A single chromosome could look something like this. And that's a shape you'll become more familiar with as we kind of do some exercises with DNA. RNA is another nucleic acid. It has just as many important functions as DNA. It tends to be a little more transitory, meaning it's usually made for kind of short-term uses, especially messenger RNA. Messenger RNA or mRNA is going to be made in the nucleus where the DNA is located, and then it is going to move out in eukaryotes to the cytoplasm, where it has to interact with the ribosome to make proteins. So messenger RNA is an integral part of protein synthesis, as is tRNA. So these 2 are involved in the process of protein synthesis. So is the final type of RNA, ribosomal RNA, but it's more functional. It is physically a part of the ribosome. So a ribosome is made up of rRNA and protein bonded together to form that structure of a ribosome. tRNA, on the other hand, sometimes people will draw it like this. There are a couple of different ways that you can show it. It's got a couple of important parts to it. This part is called an anticodon. It's got 3 nitrogen bases sticking out, and then it's got an amino acid on the other end, and it acts as the actual translation unit between the language of nucleic acids and the language of proteins. So it is also very directly involved in protein synthesis. RNA, while it's usually single-stranded, can actually form some double-stranded regions when you have complementary sequences that hydrogen base together. So notice that you have this region folded and forming a double-stranded region, this region as well. This can give you more complex shapes to your RNA, especially with rRNA or tRNA. If you look at tRNA, these sections right here, these sections, and these sections are all double-stranded sections of RNA that help form that 3-dimensional structure. So let's go back and talk about DNA replication, very important part of cell cycle. What ends up happening is, since DNA is complementary, you can use one strand of DNA, which is called the template strand, as an original, and then make a copy from it. DNA is replicated semi-conservatively, meaning that each new DNA molecule is going to be semi or half of the original DNA molecule, and it's going to have one newly synthesized strand. This is important because your cell has ways to mark the original strand. It'll add like what are called methyl groups, so CH3s or something like that, to the old strand to mark it, and then it can go back and proofread, double check, and make sure that the new strand of DNA that's been synthesized has a matching sequence to the old strand. So this all starts out when an RNA primer is laid down. That is going to be created by an enzyme called primase. It is going to set this small RNA primer in place, which is necessary for DNA polymerase to later attach and start adding in new nucleotides. It'll later have to be replaced by DNA. So DNA replication involves a lot of enzymes, and it's a fairly complex process. You have a structure called the helicase. Helicase is going to unwind the DNA double helix. You have primase, which is going to add an RNA primer. Then you have a DNA polymerase molecule, which is going to come in, and it is going to synthesize new DNA. It's going to add on complementary base pairs. You also have a few other enzymes involved. Topoisomerase is necessary to help kind of relieve the pressure from unwinding DNA. These single-stranded DNA binding proteins are also necessary to help keep the DNA from collapsing back on itself before it is read and synthesized. You have this structure called the replication fork, which is where helicase is unwinding the two strands and where you're going to have the actual replication happening. Notice we have two different means of synthesis, leading strands and lagging strands. The leading strand is synthesized continuously in one whole piece, but the lagging strand, since it is kind of running backwards to the normal direction DNA is synthesized. Remember, it's synthesized from 5' to 3', but if you were trying to do that continuously, you would be having to synthesize 3' to 5', and that just doesn't work. That would be like you trying to write backwards, which you might be able to eventually do, but enzymes are not as flexible as we are. So what happens is it is synthesized in chunks. As a section of DNA is exposed by unwinding the DNA, you're going to have a primer laid down and you're going to synthesize that segment of DNA. Then as more DNA is exposed, you're going to have a new primer laid down, and then you're going to synthesize more DNA. So it's going to be synthesized in pieces. So these are just kind of definitions for you guys of topoisomerase and single-stranded binding proteins, but we talked about their function on the previous slides. Leading strands and lagging strands are, like I said, synthesized differently. The lagging strand is synthesized discontinuously, which leads to the production of what are called Okazaki fragments. Each of these Okazaki fragments starts off with an RNA primer. And then in these gaps right here that you see, that I'm indicating with boxes, those are breaks in the sugar phosphate backbone that then have to be joined by an enzyme called ligase that fixes those breaks, forms a covalent bond to make a single continuous sugar phosphate backbone. All right, that is the basics of nucleic acid structure and DNA replication. Protein synthesis will be the topic of our next lesson.